Welcome to MOOC course on Introduction to Proteogenomics. We have finished couple of hands-on session about how to use mass spectrometry data analysis using Protege software. You also gone through many of the basic concepts of looking at mass spectrometry data. Today we have another distinguished scientist, Dr. Debashish Das, who is a professor at CSIR IGIB Institute in Delhi. Dr. Das is going to talk about integrative proteogenomics approaches in understanding of human proteoforms. As you know, proteoforms are various modified forms of a protein molecules after different modifications in a living system. In this lecture, Dr. Das will talk about the importance of proteoforms in human system by taking a reference of a research paper by Dr. Rudy Ebersold and also by sharing some of the work done in his own lab. Dr. Das will also provide you information for some of the repositories available to look at the protein proteoforms. So, let us welcome Dr. Debashish to tell us about integrative approaches, what proteoforms are and their role in clinical biology.
The topic I chose today to share with you is an integrative proteogenomics approach to unravel human proteoforms. So, this title has three terminology which needs attention. One, of course, proteogenomics, the conference is on that. The second one is proteoform. We will try to understand what are these proteoforms I am talking about, and the third is the integrative approach, and the integrative approach is what I mostly work on, and so my major thrust will be the approach by which we identify the proteoforms. As you can see, very nicely covered in Nature Methods 2013 and later on in 2014 expert review on proteomics. Proteoform actually all the alternate forms of the protein which can arise because of alternate splicing, the mRNA and any variation in or the translational errors, all of them put together, or even the amino acid modification, all of them put together can lead to generation of a variety of proteoforms of a same protein. Now, all this while we have been talking about the missing protein. So, we had a catalog of human protein and we were looking for what are the proteins that has a transcript evidence and do not have a protein evidence. Now, from there on we move to identify all the proteoforms. So, there are expected to be around 1 lakh proteoforms. The number can vary, but this is what people guess, and which are those proteoforms that are active, that are functional, some of them are involved in diseases. So, discovery of this proteoform actually can give better understanding of the functioning of the tissues. So, Rudy very nicely covered this area in Nature Chemical Biology this year 2018, that how the proteoforms will be implicated in the future of biology. So, we need to understand first of all where are these proteoforms, where are they expressed, and what are the possible roles of these proteoforms we need to understand. But before we go there, we need to understand where are these proteoforms. A tissue wise atlas of the proteoform need to be done, and that is what the research topic on which my student Anurag, who is in the audience, he works on, and some of the work that I will be presenting is done by him. Now, some basic couple of slides I have kept for those audience who are new to this area. So, how do we detect first of all the peptides in a shotgun proteomics experiment, mass spectrometry? The left hand side, which I never do, the experimentalists do, protein extraction, digestion, injection into the machine and then getting the spectra. And my lab starts from here and do the right hand side job. So, creation of a database, which is very important. Unless we create the right kind of a database, we will not get the answer. So, this database creation, I will little bit delve into this. Theoretical digested peptides generation. So, this is a rule based, so there is nothing much here. And then the peptide simulated fragment generation, which will give us theoretical spectra. Now, matching of the theoretical spectra with the experimental spectra and thereby giving a score to this is what is needed. So, here is an example. There are 3 words I have written here: allergy, gallery and largely. They are constituting of the same alphabets. So, the amino acid composition are the same. So, but the peptides are different. So, in that case the answer lies in the MSMS, the fragmentation pattern of these the fragments that will generate from these 3 peptides, and a matching will be done, peptide spectrum match scorer. So, this is different for different algorithms, how Mascot works, how Sequest works, how Tandem works. All these scorers will differ in their way of giving a score to this, but however all of them will get some of the other score. So, now it depends on us or on the method to say who has passed and who has not. So, what is the passing score here? Nobody knows, and in fact that can be a debate here. To do that, people take this approach. They create a decoy database. A decoy database is a falsified database, a database which do not contain the natural proteins. So, the proteins read from right to left maybe, or randomized shuffled sequences and the target sequence. And when you draw a threshold, that threshold actually divides the true positives from the false positive. To do this, there are many such search engines are available. Most of them you are familiar. The one that MassWiz is developed in my lab, and all others are also available in the domain, and many more also have come across. So, what they do is that they give us a lot of peptides that are identified with a score, but then some of them are positive, some of them are negative. Whereas, in a decoy database we know for sure that the peptide that we have got and the score distribution we have got are all false. Now, a comparison between this target and the decoy and a proper threshold will let us know what is exactly the passing score. So, what is that we generally say the false discovery rate? The false discovery rate is generally calculated like this: every 1 incorrect in 100 correct. So, 1 in 100 is the false discovery rate, or 5% false discovery rate, 5 incorrect is allowed for 100 true positives. So, this was again very nicely covered by Nesvitsky in Journal of Proteomics 2010. You people can go and read this article, very nicely written, which says that all the target, all of a sudden a decoy comes. So, FDR is number of decoy divided by total number of peptides that we have identified from the target. So, this is how we get to know the FDR at the peptide level, at the PSM level. But then the next challenge would be to identify the protein FDR, protein level FDR. We will see. Now, the decoy database, as I told, is a reverse or randomized sequence alteration of the original sequence, so that we can keep the amino acid composition intact, we can keep all other properties of the protein while shuffling all these amino acids. And this is already I have covered. So, this is 0% FDR, when no red is above this score, no red is there, but this is too purist an approach. So, what we generally do, we reduce our bar in such a way that we accept allow few reds and get some more greens into our search results, and that is how the PSMs are obtained, and from there the story begins. We get peptides, we match these peptides back to the proteins, and from this the protein we infer what are the proteins that are true for our experimental data. So, there are 2 ways one can do FDR calculation. One is a concatenated search, another is separate. In concatenated, what you do, actually you merge the target and the decoy into one database, whereas in a separate search you search in the target separately, you search in the decoy separately, and then you apply this formula whatever I just now told, FDR is a ratio of decoy to target. Now, what happens in the case of, so whatever just now I said is very generic, those who probably did not understand the FDR, so that is how I narrated in a brief manner. In proteogenomics case, what happens is that you take the genomic sequence, you translate computationally and create the protein sequences, and thereby you inflate the database size. So, the database when it is inflated, the chance of FDR enormously increases. Earlier we used to search, let us say, only the known protein from Swiss prot we took and search our mass spec data in a limited number of proteins. In case of proteogenomics, I take all the theoretical ORFs in case of prokaryotes or translated transcriptomic in case of eukaryote and inflate the database size. Now, when we inflate the database size, the chance of false discovery increases. How? Supposing you are looking for a place, let us say Bhubaneswar, and I have given you only the map of Orissa and you are searching Bhubaneswar. So, the chance of once you get it, it will be correct. I give you a world map and then ask you search Bhubaneswar. So, there are chances that by chance you will get another city with a similar name with one letter change here and there, and then you start getting confused which one is the right one, which one is the wrong one. So, this is the same thing happens as soon as you increase the database size, your false discovery rate increases, and then you need to do, but you have to do proteogenomics. So, database size has to increase. So, you have to find out way how do I limit my false discovery rate even though I search in the larger database. Any suggestion? I need to increase my database size because I have to do proteogenomics, but at the same time I want to reduce my false discovery. I want to reduce my false discovery. What do I do? What is the way out? Answer would be there in the next slide, but just for interaction sake. You can be wrong, no problem, but still participate. Select the peptides with very high score, that means a purist model. So, the chances of being wrong will be less, that is one way, but you will definitely lose many other correct peptides in the process, that is another one way definitely. Any other way? Can replication. Okay. So, two different experiments, you look for the same peptide being identified. So, you force me to go to the biologist and ask them to do a replicate study. Of course, that is a good idea always, but we can always increase our search engines. We can use different search engines and take their results and hope that multiple search engines will not simultaneously fail in giving you a wrong result. So, this is one way we thought of in the computational lab, where can we improve our search result, maybe include results from multiple search engines. So, what we did, we created a pipeline which will do all this process automation, at the same time take result from various search engine, one in house, but others from other sources, and take results from all the search engine and then try to analyze the data and hope that the chance of being wrong is less. What happened when we did this? We got a scenario like this. Another question coming your way. Now, world is not that simple to me. All search engines gave different results. Now, what do I do? Whom do I trust? What do you do if you get results like this? Same experiment, same database, search parameters being same, the search engines are giving you different results. And you are a PhD student and you have to take a call now. Take the overlapping. Somebody said consensus from here. I think both of you are telling the same thing. I mean, if you are agreeing to this idea, no need, but any other radical thinking idea is coming? elimination on what basis? On their scoring value. Okay. So, this is one different idea is coming. Compare their scores and then eliminate the weaker ones. Now, the problem with me is that a student coming out of IIT Bombay, if he gets even 70% mark, is a smarter student, and a student coming out from unknown university from a remote place is getting 95%, but still is not a smart student. Now, our evaluation processes are not streamlined. So, relying on the score that the student has got was not probably a smarter way of doing that. But of course, we are thinking in that line, can we rescore them? Can we create an entrance examination for all of them to reappear and come through that entrance examination again? So, something in that line we are thinking, but our first thought was whatever you suggested, take the consensus one, easiest probably and little bit safest, but definitely not the smartest. So, we went ahead with this, took all those peptides that were identified with 2 or more algorithms and made our story, went ahead, published, and that is how generally under pressure you do. But then we were not happy with the way as a computational biologist we handled the problem. So, we started observing what is the behavior of this FDR. So, look at this curve and try to understand that how the FDR is behaving as the score is reducing. So, the score is reducing to the right and the FDR axis is in the y axis, and the FDR was 0, all of a sudden a red bullet comes, the FDR shoots up, and then more and more greens are coming, the FDR is going down, and then another red hit comes and then it goes up. So, this is the function by which the FDR is jumping up and down. Now, the problem with us is that a peptide which is identified with a higher score had higher FDR than a peptide which is identified with lower score had lower FDR. This is not acceptable. How can you have a person having higher score and still has high false discovery rate? So, what we did, we created a step function and tried to join through a linear line at the base of this next FDR line, and this was fairly okay with us because still the FDR is same for this peptide as well as this peptide, for this score as well as this score, the FDR is same. But the best was when we joined these points through a linear regression lines, and now we have a curve which is going upward. As the score goes down, then you get the FDR is going upwards. What was interesting is that all the methods, irrespective of it is mass-wise, sequest, UMSA, tandem, whatever you take, this green line, this behavior of this green line was preserved. So, it was easier for us to create a cutoff for FDR score and then use that FDR score for all the methods and choose the peptides. So, what we did, we took the E value, P value score, whatever we had, the evaluation parameter, the metrics we had, and then applied on all the methods, and at a given cutoff for all these algorithms, we selected those peptides which is following that cutoff criteria. So, that part is over now. Now, multiple algorithms, search results integration, and then getting a pool of peptides from there is what we could achieve. But the main problem was to identify proteoforms. So, how do I now get the proteoforms? We have created a translated transcriptomic database. We have now created multiple algorithms and then rationalizing the results from multiple algorithms. Now, can we have an end-to-end solution for a mass spectrometry person coming with a data and do a proteogenomics end-to-end solution? So, to do that, we needed a bridge for this, and we constructed this bridge. So, we named it as GenoSuite. Rest of the talk will be little bit boring because that I will beat my own drum. This is what we have done, but nevertheless just see that what we have done. For prokaryotes it was much easier for us. 6' translated database creation, it was cakewalk, and we could get the genome reannotated with new ORFs identification. But whereas for eukaryotes we had lots of difficulty because we had to create the 3' translated transcriptome and then incorporate all the possible alternate splice forms into our proteome. To do that, we had created this pipeline by taking the best of tools available elsewhere. So, we did not write any of these codes. So, we took the SRA, Trigonometric, STAR, cufflinks, whatever was available for analyzing the RNA-seq data. All that we require is our set of protein sequences which represent this transcriptome, and this is our search space. And then using that, using multiple search engines, we wanted to get the peptides, and from peptides infer the protein. I am using the word infer because it is a bottom up approach. What we get are actually the peptides, but what we pose that as if we understand the protein now, we know which protein was there. So, from these blocks we infer what are the proteins that we probably would have got. From the first part, the prokaryotic story, we published several papers in which using this particular pipeline, we could identify new translated regions in Shigella flexneri, in Bradyrhizobacterium japonicum and Methanobaacter extracans. And if there are students in this audience who are computationally oriented and want to do something, so here is some low hanging fruit for you. As a researcher, what you can do, take mass spectrometry data from the internet, take genome proteome data from the internet, use some of these tools and then start re-annotating the genome using the experimentally available mass spec data and the static information of the genome data, and you can do wonders by sitting at a lab, in a place with a computer and internet connection. You can do all these things. So, some of these also I could get it done through the trainees who come to my lab, and we could re-annotate the genome, identify novel translated regions. So, for that the resource is available already. So, browse the internet, you will find many places where you get mass spec data, and from there you can download data. What we have done, and for our purpose, since the prokaryotic part is already whatever we wanted to do, we wanted more challenging jobs. So, we wanted to go to human proteoforms. So, we looked at the resources, and with lots of effort and difficulty we could arrive here. Although it looks pretty easy, simple to you, from the resource it was difficult for us to identify which are those projects that will give us brain specific mass spec, blood specific, lung specific, and different tissue wise mass spec data, because you cannot download the entire data and then re-annotate and then segregate. We wanted to create a pipeline which will go talk to the PRIDE database, massive database and other resources, and once you type brain, it will fetch all the brain related mass spec metadata and give it to you for the analysis. So, for that we created this human tissue scape, and this is the statistics of the PRIDE project, where you can see how many number of projects we have per tissue, and then we grouped them on the basis of their group identification, DOI, or the publication date when it has come, and that is how we could group them. As you can see, it took about several months for us to analyze tissue by tissue where are the proteoforms, and after doing the analysis we realized, oh, this is not the human tissue I was looking for, it is just a cell line. So, lot of back and forth we had to do. A lot of lesson we had to we learned while doing all these things, that it is not that straightforward. You type lungs and you get it, and then you get that is some other cell lines which people have already done the analysis. So, right now my student Anurag, who is here, he is focusing only on brain, and this is only a handful of data sets that we have analyzed using this strategy of UGenoSuite, using Nextplot, Swissplot and GenCode. We could identify several proteoforms in various tissues, and all these proteoforms now have been okay. This is interesting. Am I run out of time? I need another 5 minutes. Okay, sorry. This is something very interesting. Another puzzle which is yet to be solved in a computational pipeline manner. Otherwise, right now a lot of involvement is required. See, these are the isoforms, these are the peptides. It was much easier for us only when we had a unique peptide for that particular proteoform, which is not shared with other proteoforms. So, these proteoforms could be identified, and then they have been put into the database. These are all the proteins, their function, their gene names and number of proteoform each of them has. For example, tau protein, if you look into it, then you see that these are the various proteoforms of tau available, seen in how many different projects, how many distinct peptides were identified in that particular protein. And but then this came. What is this? These are different proteoforms of tau, these are different peptides, and this puzzle is for you, not for me. Which proteoform is present? If you see a data like this, what would be your answer? Somebody took a stand first, peptide 3, is that what you are saying? These two?
So, in this case, what we have to see, like suppose if I am talking about peptide 5 and 6, so peptide 5 has 3 proteins.
The peptide 5 is mapping to 3 different proteoforms.
Yeah, proteoform.
And 6 is mapping to 2.
Yeah, so 2 we will leave, but peptide 5, that is tau F, we can say this protein is there.
Tau F is there.
Yes.
Based on this much data, you will say tau F is there. Okay, so there is no simple solution to this problem. Even the answer that I will give you, we can spend another half an hour debating on that, but we chose it like this. We mapped all these peptides onto their transcript. As you can see, for every match, we say that evidence for D, tau, fetal E, F, but not G. For the pink peptide, that is this one, these are all possible, not these two possible. Now, these two possible. For the green peptide, where are the green one? Yeah, it is here. For this green peptide, evidence for this, but not this. So, this but not this but not this, through a series of statements like this, and also from other, because this could not be shown in one single slide, I broke them into another slide. So, evidence of tau D and fetal tau, comparing all of them together, we just given an answer that probably these are the 3 proteins most likely. I mean, the answer is most likely these are the 3 proteoforms which are there in my sample. But as I told you very clearly that we can again debate for another 1 hour or overnight on this, why this is possible, why that is not possible. So, we have to go back to the data, and then you can see, look at the peptides. This particular peptide is a unique peptide which clearly says after E the AEE comes, which is very difficult to read from here. So, which will tell that only one proteoform is possible. Other proteoform cannot explain such a separation of the peptides.
So, I hope from this lecture of Dr. Debashis Das, you got a glimpse of how one can process the proteomic samples and prepare a database to facilitate mass spectra data understanding and analysis. You also learnt about preferable limit and role of fast discovery rate in mass spectrometry data analysis. We have also learnt about the hurdles which are related to the multiple algorithms available for data analysis. Dr. Devashish explained about the possible ways to eliminate and how to select the proteins from differently used algorithms. We have also learnt about various sites for database search like UniProt, NextProt and GeneCode to make customized database for the study. Use of hubs prot for accessing already reported proteoforms of a gene could be another valuable resource. So, the next lectures we are going to shift gears, and now Dr. Mani will take you to workflows of automated data processing. Thank you.